Animal cloning. Reproductive cloning is the process of making a full living copy of an organism. Reproductive cloning of animals transplants nuclei from body cells into eggs that have had their nucleus removed. That egg is then stimulated to divide using an electrical charge and is implanted into the uterus of a female.
Hello, today we will do a process of cloning. Firstly, we need two types of sheep. For the first sheep is adult sheep, and for the second one is adult female sheep. For the adult sheep, a donor cell is taken out, and for the adult female sheep, an egg cell is removed. For this egg cell, the nucleus is need to be removed. Move to the next step, which the two cells is fused together by using the electric shock.
The fused cell begins dividing normally and produce embryo. The embryo is placed in the uterus of a foster mother, and finally, the embryo develops normally into a lamb, and its dolly comes to the world. Yay!
Let's learn about transposable elements. Transposable elements make up approximately 44% of the human genome. Mutations caused by transposable elements are linked to cancer and other diseases. First, the retrotransposon gets itself copied. There are two genetic languages, DNA and RNA. You can take the DNA as a main blueprint of life, while RNA is a messenger of life, carry the blueprint. The retrotransposon gets the help of enzyme RNA polymerase to be transcribed or copied into RNA. But the problem is, if the retrotransposon is to be inserted into a new region of DNA, it can be RNA and it can be DNA. So the RNA is reverse transcribed into DNA using reverse transcriptase. Now the copy DNA must be pasted into new part of genomes. Please bear in the mind that it's not yet fully understood how the copy DNA is pasted. The target site is where the new copy is inserted in. The enzyme integrase coats the target site, making target breaks or coats set opposite end. The retrotransposon finally pastes in. The remaining gaps are required by replicating the DNA on opposite site using DNA polymerase. Another enzyme called DNA ligase glue the segment together. The DNA transposon is cut up by the enzyme transposase. It is shaped into the hairpin structure to be pasted into the target site. The transposon attack and insert itself into the target site. Do you remember our friends DNA polymerase and DNA ligase? They are back again to fill the In summary, the retrotransposon is copy and paste into target site through the processes transcription and reverse transcriptions. The DNA transposon is cut and paste it into the target site by being changed into hairpin structure.